Hello everyone, my topic is ribosome active centers and its biogenesis in eukaryotes. So, in synopsis, the index, which consists of introduction, ribosome structures, active centers in ribosome in protein synthesis, and last one, biogenesis of ribosome in eukaryotes. In the introduction, the ribosome, which is a complex molecule made up of ribosomal RNA molecules and proteins that form a factory for protein synthesis in cells. The scientist George E. Palladay in 1955 discovered ribosomes and described them as small particles in the cytoplasm that preferentially associated with the endoplasmic reticular membrane. So he found that in the ribosome, which is a tiny particles which can be seen in the cell cytoplasm as free form and also in the associated form, which is associated with the endoplasmic reticulum. That structure is called rough endoplasmic reticulum as RER. So ribosome have especially they have two main functions. That one is decoding the message. Where from it comes from the DNA and in the form of mRNA. So they decoding the messages from the mRNA and also formation of peptide bonds between the growing amino acid chains. These two activities reside in two large ribonucleoprotein particles RNPs of unequal sizes, the ribosomal subunits, which is called the ribosomal subunits. Okay, each subunit is made up of one or more ribosomal RNAs, that is rRNAs, and many ribosomal proteins. We have already told that. So the ribosome are organelles, cellular organelles, which is located inside animal, human, and also in plant cells. They are situated in the cytosol, some bound and some free floating to the membranes of Cars endoplasmic reticulum. They are utilized in the decoding DNA, that is deoxyribonucleic acid, to protein, and no rRNA is forever bound to the RER. That is, they release or bind as directed by the kind of protein they proceed to contain. So, in an animal cell or a human cell, there could be up to 10 million ribosomes, and numerous ribosomes can be connected to the equivalent mRNA strand. This structure is known as polysomes. The polysomes can be seen only in the eukaryotic cells. So here we can see the structure of a ribosome in the cell cytosols, and some ribosomes we can see here only the attached forms. That is, in the first picture, the endoplasmic reticulum contains small black tiny dots. These are the ribosomes, and they in attached form, that is RER form. So in the second picture, we can see the large subunit and small subunit. The large one is in orange color, and the small one is a pale blue or white color. So inside the large subunit, there is a tRNA, a T-shaped RNA. It's transfer RNA, and which is growing an amino acid chain, and there is a mRNA, which is between the large subunit and small subunit. Especially the mRNA is only we can see the mRNA inside the small subunit. Okay, so the next picture is eukaryotic polysomes that consist of many ribosomes which is attaching like a bead on string form, and the string is mRNA. Okay, and the polypeptide chains are growing from the mRNA also. So the ribosome structure, look at the ribosome structure, we can see that they are made up of proteins and ribonucleic acid in also equal amounts. It comprises two sections, that is known as subunits. One is large and one is small. We already say that the tinier subunit is the place where the mRNA binds and it decodes, whereas the bigger subunit is the place for amino acids. And both subunits comprises both ribonucleic acid and protein components and are linked to each other by interactions between the protein in one subunit and the rRNAs in the other subunit. So the ribonucleic acid is obtained from the nucleolus at the point where ribosomes are arranged in a cell. So here there is a comparison between prokaryotic and eukaryotic ribosomes. The prokaryotic ribosome is totally as 70S ribosome and the eukaryotic one is 80S. What does this S means? The S is the sedimentation rate or sedimentation coefficient. What is that is when look at this. When we centrifuge a cell or especially the ribosome structures, we can get it as a sedimentation in the rate of 70 or something near to 70. That's why we referred the total prokaryotic ribosome as 70S, that is, it is sedimentated at a rate of 70, and the eukaryotic one is more denser, I think, that is 80S. So the prokaryotic ribosome is 50S large subunit, which is consist of 5 sRNA and 23 sRNA also. The 30 subunit consists of only 160S RNA. The RNA is also is the sedimentation rate. And in the eukaryotic ribosome, the 60S subunit consists of 3 types RNA or 3 RNAs, that is sRNA and 5 sRNA and 5.8 sRNA and 28 sRNA. The 40 subunit consists of only 80S RNA. So here the structure of ribosomes, which have small and large subunit, and the mRNA is along with this. And inside the subunits there is a 3 docking sites, tRNA docking sites, which is EPA, and that is we are going to discuss about them in the future in the upcoming slides. And we can see the dotted lines, which is the mRNA binding site. I already told that the mRNA is bind, or we can see the mRNA inside the small subunit. Okay, so our main topic in the syllabus is ribosome active centers. So we are going to discuss them here. And there are mainly 4 binding sites for the binding of mRNA and tRNA. That is, in a ribosome, which consists 4 main binding sites, and also addition this extra 2 sites can also be seen, which is one is the peptidyl transferase center and the other one is translation factor binding site. Both are in the large subunit. So the first active center or first active site in the ribosome, that is M site, it is the mRNA binding site on the small subunit. The ribosome has intrinsic affinity for template polynucleotide. The polynucleotide is the mRNA, or consists of many nucleotides, that is mRNA. It is located on the small ribosome subunit, 30S or 40S. It depends on the prokaryotic or eukaryotic one. In the region of groove dividing the head, the body, and the side bulge of it. 3 proteins such as S1, S2, and S5 can be seen in the mRNA binding sites. Protein S1 occupy in the interline position on the external surface of the small subunit in the region of the neck. It has an ability in forming complex with the polynucleotide. Binding of this isolated protein S1 with mRNA results in the loosening or unfolding of mRNA secondary structures. And the S5, the next protein, is located in the 30S subunit head near the neck, and the S3 opposite to that of S5. And one of the sites, that is peptidyl transferase center, the extra 2 sites consist of this one, And PTC. It is the catalysis of peptide bond formation. There is a peptidyl transferase center in the large subunit of the ribosome, which contain two substrate binding site. One is acceptor binding site and the other one is donor binding site, which is also known as the D and A sites of PTC. Of several peptidyl transferase activity is the main and seemingly the only catalytic function of the ribosome itself. It is responsible for the formation of peptide bonds during polypeptide elongation. The polypeptide elongation is the one of main steps in the translation also. In the translating ribosome, transpeptidation proceeds between the peptidyl tRNA and the amino acid tRNA. Amino acid tRNA is the charged tRNA. It is the first step in the translation, that is charging of tRNA to bring the amino acids from the amino acid pool. In this reaction, the peptidyl tRNA serves as the donor substrate and the amino acid tRNA as the acceptor substrate. So there is an equation like form. We can see here that peptide N minus tRNA plus A minus tRNA, that is gives tRNA plus peptide N plus 1, that is a bond formation between this and plus tRNA. During transpeptidation, the PTC binds with the 3' terminal adenosine with the amino acid residue of the A site bound tRNA at the A site, and the 3' terminal CCA sequences with amino acid residues its peptide group of the P site bound to tRNA at this site. PTC located at the large subunit on its interface side under the head and more exactly in the region of the groove separating the head from the rest of the body. There have been many attempts in isolating this enzyme from large subunits that is responsible for the catalyzing transpeptidation, but however, none of these attempts have proved to be successful. So it was concluded that the enzyme may consist of several proteins highly integrated in the large subunit and that undergoes disruption in the course of protein isolation. Then estimated that L2, L3, L4, L6 and L15 and L16 have found to be essential for the peptide transferase activity. So the next site is factor binding site. So first major site is N site and the second one is factor binding site, and the peptide site is an extra site. So factor binding site, GTP dependent binding of translation factors occurs here, and it is located on the large subunit. Elongation involves the periodic binding and release of proteins EF1A, that is EFTU, and EF2, that is EFG, by the translating ribosome. Each of these proteins is bound in the complex with the GTP and their release in the result of GTP hydrolysis. So the EF1A binding and release takes place during amino acid tRNA binding, whereas binding and release of EF2 protein proceeds during the translocation of change. Also, initiation translation involves the ribosomal binding of proteins IF2, one with the GTP, and the release of these proteins as a result of GTP hydrolysis. So there is a structure with the three sides and growing amino acid chain. Finally, the termination or translation, the ribosome binds and releases the RF proteins. GTP takes part in this process as well. All these proteins interacting with the ribosome in the form of their GTP complexes appear to bind the same region of the ribosomal particles. It is likely that their binding sites in the ribosome are either identical or strongly overlapping. In any case, these proteins compete against each other for binding the sites on the ribosome and cannot be present simultaneously. That is, when one amino acid chain is bounded or peptide bond formed between the upcoming one, then they release from the sites and brings space for the next one. So there is no competition between the amino acid chains. The third site is the tRNA binding site. Here we can see that the ribosome possesses an intrinsic affinity for tRNA. A vacant ribosome can bind with any tRNA or its derivatives, that is amino acyl tRNA or peptidyl tRNA, in the absence of a template polynucleotide. The presence of template polynucleotide makes this binding specific only the tRNA corresponding to the template codon. We can see that there is a 3 sites on tRNA binding site, that is A site, B site and E site. We are going to look at this 3 sites separately and discuss them about detail. So the first site is the P site or peptide site or polymerization site, also known as donor site. P site formed by both ribosomal subunit. It is in the intersubunit space. It is binding. It is the binding site for peptidyl tRNA. It binds the growing polypeptide chain of the amino acid. At the same time, the initiated tRNA gets binded to this site. So it is the second binding site for tRNA in the ribosome. So what is the first binding site? We see them in upcoming slides. During translation, the P site holds the tRNA which is linked to the growing polypeptide chain. It is the site that is occupied by the peptidyl tRNA prior to the transpeptidation in the translating ribosome. The retention of peptidyl tRNA in the P site has an important feature. That is, it is vital that the peptidyl tRNA should not be exchangeable with the medium during translation. The medium may be in vitro or in vivo. It contrasts when the deacylated tRNA or amino acyl tRNA occupies the P site, the site become exchangeable and the tRNA may be released. It is likely that the retention of the peptidyl tRNA in the P site is due to the contribution of peptidyl residue that is anchored by the D site of PTC. So the next tRNA binding site is A site or acceptor site or amino acyl site. When the P site is filled with the tRNA, the ribosome becomes capable of binding the second tRNA molecules. This binding takes place at another tRNA binding site that is called A site. Binding in the A site is greatly stimulated by the template polynucleotide. In this case, binding is codon specific, that is on tRNA corresponding to the codon placed in the site become bound. So A site is formed by both the ribosomal subunit. It is in the intersubunit place. In any case, the tRNA anticodon should be placed in the immediate vicinity of the mRNA codon, that is in the small subunit, ribosomal subunit, whereas the acceptor and interact A site of the PTC, that is with the large subunit. So E site or exit site. It is a site for binding the uncharged or unloaded tRNA. As a result of translocation, the deacylated tRNA is expelled from the P site. However, this doesn't necessarily mean that it is immediately released from the ribosome. The deacylated tRNA after translocation transiently retained on the ribosome in the exit site. The E site, which binds the 3' terminus of the deacylated tRNA, is in the vicinity of the PTC of the large subunit, seems to contribute mainly to the retention of the deacylated tRNA after translocation. It is not clear that yet how other parts of the ribosome, especially the small subunit, are involved in the formation of E site. So the fourth site is entry site or R or T site. It is an intermediate site for amino acid tRNA located in small subunit. When the amino acid tRNA entering the ribosome, it is complexed with the EF1A, then it cannot be a substrate for transcription until GTP cleaved and EF1 is released. So bound between EF1A and amino acid tRNA blocked it here. So on the way to the A site amino acid tRNA, sit first on an intermediate entry site, also called the recognition site, that is the R indicates, or T site because the presence of the bound EFTU. Since T site is in the small subunit, the contact of the T site bound tRNA with the small subunit are very similar to those of A site bounded tRNA. At the same time, there is no direct contact with the large subunit have been detected. So E site is not a separate tRNA binding site of the ribosome, but just an intermediate position of amino acid tRNA. When it is already bound within a site on the small subunit and not bound with the PTC of the large subunit. So that is. The next one is ribosomes in protein synthesis. We all know that protein synthesis, the main part in protein synthesis is the ribosome. It is the mediator for decoding the codes in the mRNA and changes into the amino acid form, and it's really helpful, more helpful in protein synthesis. The bringing together amino acid to form particular proteins which are the important for completing the cell's activities. That is, cell's metabolism is controlled by many proteins. Protein is required for numerous cell functions, for example, detecting chemical processes or fixing the damage, etc. etc. Ribosomes can yet discovered floating inside the cytoplasm or joined to the endoplasmic reticulum. Procedure of creation of proteins, deoxyribonucleic acid makes the mRNA by the step of DNA transcription. That is it, the first step of protein synthesis, mRNA synthesis by transcription. Then the mRNA is translated to amino acid chain with the help of ribosomes. That it's the main function of ribosomes actually. So the third one is hereditary information from mRNA is converted into the protein amino acid DNA translation. It's also a part of hereditary information transmission. The arrangement of protein assembly proteins synthesis are indicated in the mRNA. The next one is mRNA is arranged in the nucleus and is converted moved to cytoplasm for the additional operation of protein synthesis. These are main functions of ribosomes in the protein synthesis. What is not the function, what is the role of ribosome in the protein synthesis? So next we are going to look the second topic in our syllabus, that is biogenesis of ribosome, especially in eukaryotes, not in the prokaryotes. So eukaryotic ribose, which is ATS ribosome, are formed of 70 to 90 kinds of proteins and 4 different kinds of rRNA, namely ATS, 20S, 5S and 5-point-S sRNA. Here the 4-1 is large and small subunit both. Their larger subunit is formed of 40 to 50 kinds of proteins and 2S, 5S and 5-point-S rRNAs. Smaller subunits consist of many proteins and also 18S rRNA. The two subunits associated during translation and they dissociated when the translation of mRNA molecule is completed. So it's a temporary structure that we can see that the ribosome two subunits are joined together. That's the temporary structures. Formation of eukaryotic ribosomes is a complex process. Many cellular regions are involved in the synthesis of ATS ribosome, especially the nucleus, nucleolus, and the cytoplasm. Its initial stage takes place in the cytoplasm and nucleus, intermediate stage in the nucleolus, and the final stage is in the cytoplasm. To begin with, the ribosomal protein are synthesized in the cytoribosomes. synthesized on the site of ribosome and pre-rRNA in the nucleus. So it is the overall picture of what is going to be we study in the following slides, that is the biogenesis of ribosome in short. The 45S rRNA, which is the pre-rRNA, are transcribed from the genes on the nuclear organizer region of chromosomes, where we can see that the nuclear organizer genes are responsible for the transcription of these rRNAs, and the 5S rRNA from the genes outside it. In the second stage, all the pre-rRNA move to the nucleolus. Here the processing of pre-rRNA to rRNA and binding of rRNA with the protein takes place. And from ribosomal subunit also. In the last stage, these subunits are transported to the cytoplasm, where in the optimal level of Mg2+, that is magnesium, in the cytoplasm, they bind together and form ribosomes, a single unit. That is the time of translation. The nuclear organizer region is specific chromosomal portion which govern the formation of nucleolus. It is located in the secondary construction of the chromosomes. It contains the cistrons for 5.8s, 18s, and 20s, that is the large subunits rRNAs. So the cistron is the functional part of a DNA from where the proteins can be synthesized. There may be 800 copies of each of these genes. The portion of the DNA which contains these cistrons is known as the ribosome DNA or rDNA. The genes for the formation of rRNA are located in 5 pairs of chromosomes in the nucleus. Each of these chromosomes contain many duplicates of the particular gene because of the large amounts of ribosomal RNA which is required in the cellular functions. So, here are the major steps in the biogenesis of ribosomes, that is 3 events, mainly 3 events. After these 3 events, the ribosomes become they form a single structure by combining these parts. So first step is ribosomal rRNA synthesis, that is inside in the nucleolus, then synthesis of 5S rRNA and biosynthesis of ribosomal proteins. So the first step, that is ribosomal RNA synthesis in the nucleolus. Eukaryotic ribosomes have 4 distinct ribosomal RNA, 3 in the large subunit and 1 in the small subunit. We know that already. So ribosome synthesis requires the function and coordination of all the 3 nuclear RNA polymerase, that is RNA polymerase 1, 2 and 3. So the 28S and 5.S rRNA associated with the large subunit and the single 80S rRNA of the small subunit. These are transcribed by the RNA polymerase 1, and the RNA polymerase 3 transcribes the 5S RNA of the large subunit, and RNA polymerase 2 transcribes mRNA encoding ribosomal proteins. The 28 and 5.8 srRNA associated with the large subunit and the 18S associated with the small subunit are all encoded by a single pre-RNA transcription unit in the nuclear organization region, which is called rDNA. So the production of these three rRNA in a single stretch. So there is a complexity in the production of these structures. That's why there is a need for processing the rRNA. Okay, in human cells, by its transcription by RNA polymerase 1 along with the transcription factors 1 yield a 45S primary transcript, that is pre-rRNA. Along in some species it will be 40S. So we have after the synthesis of these rRNAs, we have a long stretch of rRNA which consists of uh, 28 srRNA, 5.8 srRNA, and 18 srRNA. These three are in a single stretch. So the cell can be identified this by only separating this by a spacer RNA, that is a non-coding RNA. This spacer RNA, we can see that in the uh, so for example, we say that these three rRNAs in a single ribbon or a single um, piece. So the 5' and 3' ends were capped by a spacer RNA, and also between these three rRNAs there is a spacer RNA. So the 5' and 3' end of the pre-rRNA is known as external transcriber spacer, and the spacer RNAs between these rRNAs is known as internal transcriber spacer. These pre-rRNA become processed and by cleavage and base modification into mature 28S, 18S, and 5.S rRNAs. Now they become separated. Okay, the pre-rRNA migrate to nucleolus and further processing takes place there. So we are going to look at other species like other eukaryotes. So the 45S pre-rRNA from many species showed that this DNA shares several properties in all eukaryotes. First, the properties are here. First one is pre-rRNA genes are arranged in a long tandem arrays separated by spacers, that is non-transcribed spacer regions. They ranging the length from 2 kb in frogs and to 30 kb in humans. So we have larger spacer RNAs. Second property is the genomic region corresponding to the three mature rRNA are always arranged in the same 5' 3' order, that is 8DS, 5.S, and 28S. Here the pattern for these three arrangements, these three srRNA arrangement. It is same in all the eukaryotes and from humans and also in humans. The third property is that in all eukaryotic cells, the pre-rRNA genes code for regions that are removed during processing and rapidly degraded. These regions probably contribute to the proper folding of pre-rRNAs, but not required once that folding has occurred. So only for folding it is required. So we are going to study or discuss about a new rRNAs, that is no rRNAs, that is helpful in the processing of pre-rRNA. Processing means the separation of the three srRNAs from spacer RNAs and by cleavage. So this whole process is helping by the no rRNAs. So we can say that in the pre-rRNA there contain a transcriber space region at both 5' 3' end, that is the external space transcriber space, and between the 18S, between these srRNA also internal transcriber regions. So the processing involves the series of cleavage by endoribonucleases, not the endonucleases, and the exoribonucleases for cutting or cleaving the spacer RNAs between these three sRNAs, and for cleaving the terminal or 5' 3' spacer RNAs, respectively, which remove the external and internal transcriber spacer regions, and base modification mostly by the methylation of 2-hydroxyl group of the specific ribose and conversion of the specific uridine residues to pseudouridine. We see this pseudouridine structures and methylation. What is its importance in the following ones? By the time human pre-rRNA precursors is first cleaved, over 100 methyl groups have been added to ribosome groups in the molecule, and approximately 95 of its uridine residues have been enzymatically converted to pseudouridines. The function of methyl groups here is this. The function of methyl groups and the pseudouridine is that these divine modified nucleotide may protect the pre-rRNA from enzymatic cleavage and promote the folding of rRNA into their final three-dimensional structure and promote the interaction of rRNA with other molecules. Okay, so what is the role of rRNA in the processing? The processing of the pre-rRNA is accomplished with the help of a large number of small nucleolar RNA, that is known as rRNAs. It's found in the nucleolus. So they function in associated with the specific proteins and thus form ribonuclease proteins, that is R and PS. There are about 80 different rRNA in yeast and almost twice as many in human chromosomes. These guide chemical modification of other RNAs, mainly ribosomal RNA transfer RNAs, so they are also known as the guide RNA. That is, the rNA is also known as guide RNA. There are mainly two classes of There are mainly two classes of rNA. That is CD box RNAs. They are associated with the methylation process of processed or cleaved RNAs, pre-RNAs, and the second one is HACA box RNAs. They are associated with the pseudo-urination process of pre-RNA. So the box CD RNAs determine which nucleotide in the pre-RNA will have their ribose moieties for methylation, and whereas the member of other groups called the box HACA they determine which uridines are in the pre-RNA is converted to pseudouridines. Both the group of rNAs contain relatively long stretches, that is 10 to 21 nucleotides, that are complementary to sections of the rRNA's transcript. In this case, each rNA binds to a specific portion of the pre-rRNA to form an RNA-RNA duplex. The bound rNA then guides an enzyme, either by a methylase or a pseudouridase, that is the enzymes for methylation and pseudouridation process, within the RNP to modify a particular nucleotide in the pre-RNA. Taken together, there are 200 different rNA, one of each site in the pre-RNA, that is ribose, methylated or pseudouridated. If the gene encoding one of these rNA is deleted, one of the nucleotide in the pre-RNA fails to be enzymatically modified. So what will happen? The pre-RNA will be enzymatically degraded by further mutation and changes occurs in the RNAs and the ribosome subunit. And these all changes leads to our transcription or our transfer of heredity information, and maybe have some disease or something like that. So here is the second process in the biogenesis, that is the synthesis and processing of 5sRNA. So a 5sRNA, about 120 nucleotide long, RNAs, they are encoded by a large number of identical genes that are separated from the other rRNA genes, and they are located outside the nuclear organizer region. Okay. Following its synthesis, the 5S RNA transported to the nucleolus to join the other components involved in the assembly of ribosomal subunit. So they going to the nucleolus to combine with the other subunits, other rRNAs. So these rRNA is transcribed by who? RNA polymerase 3. A gene for 5S RNA contains an internal control region, that is ICR, in its middle region, which is found essential for this transcription. To this control region of genes remain attached to a special protein that is called transcription factor 3A, or that's not 3, that is IIIA, or TF IIIA, which permits the RNA polymerase 3 enzymes to recognize the promoter of 5S rRNA genes. So, with only minor additional processing to remove the nucleotide, sorry, to remove the nucleotide at the 3' end, the 5S RNA diffuses to the nucleolus, whereas it assembles with the pre-rRNA and the remain associated with the region that is cleaved to the into the precursor of the largest unit also. So the last step is the biosynthesis of ribosomal proteins. There are plenty of ribosomal proteins which is involved in the translational process. It involves in the mRNA-dependent synthesis of proteins. Ribosomal protein synthesis involves transcription, pre-mRNA processing, nuclear export of mature mRNA, and translation or on cytoplasmic ribosomes. The mature R protein are then imported to nucleus and finally to the nucleolus. Finally, all these rRNA and protein assemble each other, forming large and small subunits separately within the nucleus. They are only combined during translation. And don't forget about that. There is a two type of protein that become associated with the RNA as it is processed. One is ribosomal protein that remains in the subunit, and accessory proteins that have transfer interaction with the rRNA intermediates and required only for processing. Among the later group and are more than dozen RNA helicase, which are enzymes. Helicase, which is enzyme that unbinds the regions of double-stranded RNA that are necessary for dislodge the SNO RNPS. The most of the ribosomal proteins of the small 4TS ribosomal subunits associated with the nascent type, that is pre-RNA during transcription, and it requires only a few more remodeling steps before it transcribed to the cytoplasm. Once the pre-40S particles leave the nucleolus, it travels to the nucleoplasm, that is, it travels to the nucleoplasm and quickly exported through nuclear pore complex NPCs. The final step in the maturation of small subunit occurs in the cytoplasm, that is exonucleolytic processing of the 20S RNA into mature subunit, small subunit 80S rRNA by the cytoplasmic 5-3-exoribonucleases XRN1, and the demethylation of two adjacent adenines near the 3' end of the 18S rRNA by the cytoplasmic enzyme DIM1. In contrast to the pre-40S particles, the precursor of the large subunit require considerable remodeling through many more transient interactions with the non-ribosomal protein before it sufficiently mature for export to the cytoplasm. So we can see here the picture of synthesis of the ribosomes and their combinations. They combine to form small and large, and the small and large subunit combine to form a large subunit. Here we can see that. So consequently, it takes longer time for maturing 60S subunit to exit the nucleus, that is 30 minutes compared with the 5 minutes for export of the 40 subunit in the filtered human cells. The multiple presumptive RNA helicases and small G proteins are associated with the maturing of pre-60S subunit. Some RNA helicases are necessary to dislodge the SNO RNPs. They are helping in the cleavage of or processing of rRNAs, pre-RNAs. So here the conclusion that taking into consideration their main function, that is the ribosome's main function in developing proteins, it's clear that a cell can't function in the absence of ribosomes. So here is the reference, that is Molecular Biology of Gene by Watson, and thank you.